Dear students, welcome to Tungala online classes. Today we will continue with level 2 questions, that is NEET level questions. So 50th question, which is not true for anaphase? Here we will have to concentrate on the stage that is anaphase. So when a wrong statement is there, we will have to identify that wrong statement over here. What is the first statement? Golgi complex or Golgi body as well as endoplasmic reticulum are reformed over here. So he says in anaphase stage, so the formation of some of the cell organelles like Golgi complex as well as endoplasmic reticulum are reformed. He is saying actually it is wrong statement. Here there is no formation of any cell organelles in this stage that take place during the telophase. So it occurs there. That's why this is the correct answer for this question. And what is the next statement? Chromosomes move towards opposite pole. Of course, in anaphase stage, so there the movement of chromosomes that take place on opposite poles. It is the right statement over here. Spindle poles move further apart. Of course, spindle poles. So there continuous spindle fibers, discontinuous spindle fibers are usually present. So they move towards they move further apart, and centromere split and chromatids separate. Of course, here the centromere that splits over here, two chromosomes that move apart in anaphase stage. It is a correct statement. So that's why, so B, C, D are correct statement. This is the wrong statement. A is the wrong statement. Hence, the correct answer for this question is the option A. Right? So we'll go to the next question. Cell plate formation starts from. Cell plate is initially starts from the formation of the phragmoplast. Phragmoplast is initially formed and later it continues to form the cell plate. So epiplast, it is not epiplast, it is not tonoplast, which is a main membrane around the vacuole, and it is not a protoplast, so which contain the cell with the membrane itself is called as protoplast, right? So that's why the correct answer for this question is a phragmoplast, right? So next question, colchicine brings about. So it brings about gene mutation, duplication of chromosomes, chromosomal aberrations, quick replication, he says. So during use of this colchicine, so this inhibits the synthesis of the microtubules during cell division, right? That is the major function of this colchicine. It brings about duplication of chromosomes as the number of chromosomes that duplicate, so number of chromosomes increases, but so there is no cytokinesis that take place. That's why it leads to polyploidy condition. So that's why duplication of chromosomes is the correct answer for this question. So next question is, if a cell has twice as much DNA as in a normal functional cell, it means that the cell is preparing to divide, has completed division, has reached the end of its life, has ceased to function. Here, when the duplication of DNA that takes place, right, which is twice as much it had, right, so it shows that if the DNA duplicates, the number of chromosomes that duplicate over here, and it leads to cell division, means so during the S phase of cell cycle, as you know, so interphase has three phases. One is G1 and S as well as G2. In these one, DNA synthesis or duplication of the DNA that take place in S phase, right, over here, and so it leads to G2 phase. There also the preparation of some of the materials for duplication of cells that occurs in G1 also. right? So it leads to cell division. Means interphase itself is called as a preparatory stage of the cell division. Hence, so after this interphase, after the DNA duplication, what happens? Cell is going to divide. Let us see options over here. So it means that the cell is preparing to divide. The cell is preparing itself for division. So has completed division. After DNA duplication, is ready to divide itself. Not it has completed its division. So that's why it is a wrong statement. Has reached the end of its life. So it hasn't reached the end of its life. Has ceased to function. So all these are wrong. First one is correct statement for this question. And the next question. Cyclin protein is required for cell cycle. Of course, cyclin is the specialized protein which is helping for the transition in cell division. So it helps for, and which other molecule is essential for completion of cell cycle. Along with this cyclin, there are some materials or the proteins, specialized proteins, that is CDKs, cyclin dependent. So protein kinase are also required for the completion of the cell cycle over here. That's why the correct answer for this one is option C, CDKs, right? And the next question. Fibroblast cells in our body are those which are arrested in. So these are the specialized cells. Fibroblasts are the specialized cells which are present in one of the tissues, that is areolar tissue and even adipose tissue. There in tissues, these fibroblasts are the specialized cells present over here, and they help in synthesis of the fibers. So that is white collagen fibers as well as yellow elastic fibers. So these are the specialized fibers which are produced from these fibroblasts which are present in, so the tissues like animal tissues like areolar tissue, right? Other tissues itself. And what is the question over here? Fibroblast cells in our body are those which are arrested in. See here, these are the specialized cells which are so changing their morphological features. So that is nothing but differentiation occurs over here in some of the cells. So a normal cell which differentiates into fibroblasts, right? And these fibroblasts, so which are branched cells which are usually producing the collagen as well as elastic fibers over here, and they are arrested usually in. So here G0 phase is G1 phase, G2 phase, yet to start division, right? There is no division occurs in fibroblast. They are already matured cells. They have so changed their morphological features. So ultimately they are arrested in G0 phase. So that's why G0 phase is the correct answer for this question. Why? Because some of the cells which get matured and metamorphosis, so as to start one particular function, so that takes place usually in G0 phase. That is why G0 phase is the correct answer for this question, right? Next question. Some mature cells cannot undergo divisions because they get mature, right? They get mature and differentiate themselves and start to do one particular function. So that's why they do not undergo next further division over here. That is the major reason. So we'll search for the same reason over here in the options. They have lost the ability to divide. One thing, their genes have become inactive. they have taken some specific functions. Of course, they start to do one particular function, right? And undergone differentiation. So, those cells which have undergone differentiation, right, as well as start to do one particular function, because of this reason, they do not undergo any further divisions. That's why, so here option C is the correct answer for this question. That is, they have taken some specific functions and undergone differentiation. Right? Next question. During meiotic division, what happens during meiotic division? Many processes, many aspects are taking place over here. So among them, among all these statements, which is the right one, we'll have to identify over here. So during meiotic division, homologous chromosomes are separated. Of course, two homologous chromosomes that participate during this cell division. So they separate during meiosis. Both of themselves separate during meiosis. This is the aspect what we have learnt already in the theory, and some of the questions related to this concept I have already discussed in level 1 questions. And so, as you know, homologous chromosomes are separated over here. One homologous chromosome is from paternal set and another one is from maternal set. So both undergo synopsis and charismata formation that take place, and ultimately terminalization leads to separation of two homologous chromosomes over here. That's why during meiotic division, homologous chromosomes are separated is the correct statement for this one. Linkage is a totally complete different concept over here. The homologous chromosomes do not segregate, he says. Of course, here segregation occurs. So that's why this statement is also wrong. All the above is also not considered here as an answer. That's why homologous chromosomes are separated is the correct answer for this question. So we'll go to the next question. The drug colchicine has inhibitory effect on the stage of the cell cycle. On which stage of the cell cycle it has inhibitory effect, he is going to ask over here. See here, already I have mentioned that this colchicine is called as meiotic poison, which arrest or which leads to arrest of the cell division or cell cycle itself, and it inhibits the formation of microtubules, that is nothing but which are going to form the spindle fibers, right? So that's why in which stage it has inhibitory effect, he is going to ask over here. So as you know, centrioles that duplicate, during what stage centrioles duplicate? Initially prophase, and ultimately the complete synthesis or the formation of these microtubules to form the spindle fibers that completes in metaphase. In metaphase, what happens? So if the synthesis of those microtubules or the spindle fibers do not have taking place, if it is not taking place, it is because of this colchicine only. That's why. So metaphase itself is one of the stages of M phase. M phase over here. That's why the correct answer for this question is M phase over here. Right? So next question. Differentiated cell arrest at which stage? Differentiated cell arrest at which stage he is asking. So once again the same concept over here. So in G0 phase, usually it arrest over here. Cell will be at resting period. And next question is the stage during which the cell decides to get specialized. Right? The cell becomes specialized and start to do one particular function. Once again the same concept is given over here. So S phase, M phase, G1, G2, right? Early G1 it arrests sometimes. Since option is G1 over here, so that's why in early G1 it decides to get specialized over here. That's why here the correct answer for this one is G1 phase is the correct answer for this question. So next question. Which of the following is an ideal for studying meiosis? So here during the study of meiosis, usually meiosis takes place during formation of microspores, that is pollen grains, during gamete formation, and in the root tip also the cell division is occurring, and during the pollen formation is also the cell division, that is meiotic cell division occurs. But the suitable one for the study of this meiosis itself is microsporangium. So in which microsporogenesis is occurring over here. The process of formation of microspores. So from its parent cell or its diploid microspore mother cell is called as microsporogenesis, and during that microsporogenesis it is better for us to study this meiotic division. That's why microsporangium is the correct answer for this question. Yes, now go to the next question. So there is no division of cytoplasm after mitosis. Then the condition will be, what will be the condition if there is no cytoplasmic division or cytokinases that occurs over here? See here, if this is one animal cell, then it has a nucleus and it has chromatin network, etc. Right? This cell initially, what happens over here? During cell division, one is karyokinases occurs and cytokinases occurs over here. Karyokinases, that is nuclear division, cytokinases, that is cytoplasmic division. Right? So these are the two steps that we have already studied over here. So what he is asking over here is, after karyokinases, if the cell is not undergoing cytoplasmic division, then what will be the result? He is asking over here. So what will be the condition? Right? One cell, one nucleus is. So what happens here? If this cell divides, then nuclear division occurs over here. Then formation of two nuclei is done, but the cytokinases is not done over here. That's why what will be the condition? One cell with two nuclei. This is the correct answer for this question. Only one cell is there, but two nuclei are there. So if a cell undergoes only karyokinases, not the cytokinases. That's why the correct answer for this question is one cell with two nuclei. What is the next question? Pick out the correct statements over here. So which are the correct statements? We will have to analyze here. So first statement, mitosis takes place in somatic cell. Of course, mitotic cell division occurs in somatic cell or the body cell. Mitosis occurs in body cell or the somatic cell. It is the right one. Right? And meiosis takes place in germ cells. Of course, meiosis that occurs in germ cells. Right? So respectively, the statement is correct over here. Both of these are correct, hence the statement is correct over here. This statement is correct. Yes, given over here, pick out the correct statement. We have picked out the correct statement. What about the next statements? Whether they are also right or wrong, you can have to analyze. During mitosis, the DNA replicates once for one cell division. Of course, for one cell division, DNA replicates once. In meiosis, the DNA replicates for twice. For two cell divisions, he says, but it is wrong. In mitosis, so there we come across interphase as well as M phase over here. During interphase, G1 S as well as G2 are the phases that we come across. In S phase, duplication of DNA take place during one cell division. It is right. But during meiosis, so the duplication of chromosomes or the duplication of DNA that occurs only once, not twice. So even though the same cell divides twice, right? So that is the aspect I have already highlighted in theory. So that's why second statement is wrong over here. And what about the third one? Let us see. Mitosis and meiosis occurs both in sexually and asexually reproducing organisms, he says. So what about the sexually reproducing organisms as well as asexually reproducing organisms? We'll analyze over here. So this is one cell. So this cell undergo mitotic division. Of course, if it is sexually reproducing organism, right? So here sexually reproducing organisms in which mitosis take place as well as meiosis also take place. Why? Because so in sexually reproducing organisms, gamete formation is quite natural one. So during gamete formation, meiosis must occur there in germ cells, right? So and mitosis occurs. See here, after the fusion of male and female gamete, this is male gamete, this is female gamete, both of themselves fuse together form a thick word zygote over here. This zygote undergoes repeated division of mitosis. So what happened here? Even though the organism is sexually reproducing, mitotic cell division also take place and meiotic cell division also take place. So, and mitotic cell division in case of sexually reproducing organisms is responsible for the growth and development of the organism over here. That's why, so in sexually reproducing organisms, both mitosis as well as meiosis take place. What about asexually reproducing organisms? There is no need of any meiotic division there. That is why, so this statement is also wrong one. The correct statement for this question is, so the first one, that is mitosis take place in somatic cell as well as meiosis take place in germ cell. Is that right answer for this question? Next one. RNA and proteins are formed differing. So RNA as well as the proteins are formed differing G1, G2, S as well as G0 phase. In G0 phase, so the cell will be always metabolically inactive over here. Metabolically inactive, right? So the cell will be metabolically inactive here, right? The synthesis of RNA as well as the proteins required for the cell which is going to get ready for cell division, that takes place during G2 phase, right? And next question, variations appear during. At what time the variations occurs over here? Already I have mentioned in theory. So what is that? So variations are nothing but the differences among the individual organisms of the same species. The differences in the characteristic feature, morphological characters among the individuals of the same species are called as variations. So those variations are occurring during. See here, in meiosis, there we come across meiosis 1 as well as meiosis 2. In meiosis 1, there prophase 1 we come across. In prophase 1, there we come across 5 stages LZP, DD. So in that one, L leptotin, zygotin. So but in pachytene, what happens? So there the crossing over occurs over here. Crossing over. This crossing over leads to change in the genetical characters due to exchange of the chromatids between two homologous chromosomes. See here, this is one homologous chromosome, this is another homologous chromosome. There, what happens? The exchange of the chromatids take place. So where the crossing over point is there, from that point there, this chromatid cuts over here, this chromatid cuts over here. The combination of the genes in both the chromatids that change, that is called as genetical recombination, it is called as. And as a result of this genetical recombination, what happens over here? So there, the variations in the genetical characters that occurs. That's why the correct answer for this question is option B, crossing over is the correct answer for this question. Next one, during mitosis, endoplasmic reticulum as well as nucleolus begins to disappear at, right? So during mitosis, so these nucleolus, nuclear membrane as well as some of the cell organelles, they start to disappear. So at early metaphase he says, late metaphase he says, early prophase he says, late prophase he says. See here, the nuclear membrane as well as the nucleolus start to disappear in the late prophase stage. In the early stage, disintegration of the nuclear membrane, right, starts, and ultimately the disappearance that takes place in late prophase, and total disappearance is seen in case of metaphase, early metaphase over here. So there the chromosomes are left into the cytoplasm directly, right? That's why the correct answer for this question is late prophase. Option D, late prophase is the correct answer for this question. Next one, how many chromosomes will the cell have at G1, S as well as after M phase respectively if it has 14 number of chromosomes in interphase? If in interphase during mitotic cell division, so if 14 number of chromosomes are there, then automatically the same number of chromosome is maintained over here in this type of a cell division over here. So that's why the correct answer for this one is in G1 14, in S 14, 14. So it is the right answer for this question. In all the stages there we come across same number of chromosomes. That's why, so option B is the right answer for this question. Crossing over, next question is, crossing over is also an enzyme mediated process, and enzyme involved in is called as, what is the enzyme involved, enzyme complex involved in this called as? This is the question. It's very simple question as we have studied. Once again, meiosis 1. In meiosis 1, there we come across prophase 1, metaphase 1, anaphase 1 as well as telophase 1. In that one, prophase 1 is the longest period activity. So where LZP, DD are the 5 stages that we come across, right? So there in L, lactogen stage, the chromosomes are looking, they are arranged as a bouquet stage it is called as, and Z in which the homologous chromosomes, one set paternal, one set maternal set, so they start to overlap on each other so as to form synapses over here. And in packaging, the synapsed chromosomes, they cross over there. Crossing over occurs over here in packaging, right? So in diploid, there schemata formation, dikinases, terminalization. All these aspects that we have highlighted every time in the questions. What is the question? Crossing over is also an enzyme mediated process. Of course, there is an enzyme needed for crossing over, as you know, two homologous chromosomes are lying very close to each other, and non-sister chromatids, they cross over each other, and there we come across one of the complexes what is formed, enzyme complexes, recombinase nodule is formed out here. What is formed? Recombinase nodule is formed in between two non-sister chromatids, between two homologous chromosomes, and this is an enzyme complex, what we call it as. It is an enzyme complex which contains some of the enzymes like endonuclease as well as ligases. So endonuclease that help in cutting the chromatids, right? And exchange of the chromatids that take place between two non-sister chromatids of two homologous chromosomes. So as a result of the activity of these enzymes that take place, of course. So it contains enzymes like endonuclease as well as ligase, of course. So these two form the recombinase it forms. Then the correct answer for this question is, so we should not highlight endonuclease as well as ligase. So recombinase is the correct answer for this question. Even though these two enzymes which are present in the recombinase nodule, so we should not correct these one, we have to correct recombinase one, right? It is an enzyme complex, so that helps for crossing over here. So next question is, what is incorrect about G naught? In G naught stage, as already I have mentioned, so where the cell is inactive in nature, metabolically inactive, there is no growth at all. So there the cell is going to differentiate itself, right, and start to do one particular function. That is also possible there. Then what is the wrong statement about this G naught? Let us see. Option A, cell metabolism continues in G naught. The metabolic processes are held over here, so that's why this is the wrong statement for this G naught stage, where the cell is metabolically inactive, but he says metabolism continues in the G naught, he says. So metabolic activity is not taking place. Cell growth occurs in G naught, of course. Biocatalyst help in exit G naught. Meiosis occurs after G naught. So this is the correct statement for this is the wrong statement for G naught. That's why the correct answer for this question. Next one. Plane of cell wall formation in a dividing cell is determined. So what determines the plane of cell wall formation? Usually cell wall is present in the plant cell especially, and what decides the cell wall formation over here? So let us see the options. Microfilaments, he says, and microtubules, he says. Golgi complex, endoplasmic reticulum. Golgi complex as well as endoplasmic reticulum, these are the cell organelles. They do not decide the formation of the cell wall over here. So there, what decides then? Microtubules. These microtubules are forming the spindle fibers. Tubulin is the protein that forms these microtubules, and the microtubules form the spindle fibers, and these spindle fibers decide the cell wall formation. Why? Because the spindle fibers what are organized at the equatorial plate, they gather themselves, and there the gathering of some of the vesicles of Golgi complex also take place, that leads to the formation of the cell plate, phragmoblast initially and cell plate later, and that form the middle lamella as well as cell wall. So that's why microtubules are forming the spindle fibers, which decide or which determine the cell wall formation, the plane of cell wall formation. That's why microtubules itself is the correct answer for this question. So next, we shall go to the next question. In diploid cell, at which stage of a cell cycle the amount of DNA is doubled? Repeatedly we have gone through this one. It's a very simple question. The answer for this question is S phase, where the duplication of chromosomes of the DNA that takes place over here. So next question is, the enzyme recombinase is required in which stage of meiosis? Already this question is discussed over here. Recombinase nodule formation that take place in packaging stage. Since in the packaging stage only one process, that is crossing over, occurs over here, this results in formation of variations in the individuals of the same species. So right, that's why the correct answer for this question is, he gave here zygoting. In zygoting usually, so it is the second stage where the synopsis that occurs. In diploting, chiasmata formation that occurs. In dikinases, terminalization occurs. In packaging, of course, so crossing over occurs. That's why recombinase is an enzyme complex required for packaging stage. That's why the correct answer for this question is packaging over here. So these are some questions I have solved for you, and later we'll discuss some of the level 3 questions now. Next, level 3 questions are here. Lamprecht chromosomes are found in. Already I have told you, Lamprecht chromosomes are a kind of joint chromosomes which are usually seen in oocytes, right? So they are usually seen in the protein stage, and so these chromosomes are usually seen in the protein stage. So is the right answer for this question. And the next one, colchicine arrests which of the following stage of cell division? Colchicine is one of the meiotic poison which arrest or which stop the spindle formation. So that's why spindle fibers are completely formed during the metaphasic stage. Hence, so the metaphasic stage at which the cell division is arrested. That's why the correct answer for this question is metaphase. And the next question is, dyad chromosomes are observed differing. So dyads as well as tetrads are the type of chromosomes that we come across over here, but the dyads are usually seen in, he says, prophase 1, metaphase 1, anaphase 1, as well as meiotic anaphase. So dyads are usually seen in anaphase 1. So when you see the diagram of dyads there, so in anaphase 1, so you can identify. Next one, which one is the mitogen? Mitogen are nothing but they are a kind of substances help for cell division. So cytokinin is one kind of what? Mitogen. And ECF is also one kind of mitogen, and so platelet derivative spindle pores is also one kind of mitogen. These are the different types of mitogens that we come across here which stimulate the cell division. So that's why the correct answer for this question is all the above is the correct answer. We have discussed all these things in the theory in detailed way, right? And the next one, double metaphasic plate is observed in. As you know, so there two line of chromosomes are arranged in one of the stage. So that stage is what? So metaphase 1. In metaphase 1, the two lines of chromosomes are arranged so parallel to the equatorial plate. Hence it is said to be as metaphasic plate, and metaphasic plate is looking to be as a double. Hence, so that double metaphasic plate is observed in case of metaphase 1 is the right answer for this question. Next one, in onion root tip, during mitotic metaphase, the number of kinetochores he is asking. The number of kinetochores for each chromosome is 2, right? On either sides of the centromere. That's why the double the number of chromosomes you calculate yourself. So in onion root tip, the cells have 16 number of, that is diploid cells have 16 number of chromosomes are there. If 16 number of chromosomes are there, automatically the number of kinetochores is double of this one, that is 32. Hence, the number of kinetochores in mitotic metaphase is 32. Hence, option C is the right answer for this question. Next question. 79th question. It is meiosis 2 is similar to mitosis. These are assertion as well as reason type of questions. So we must be analyze all these statements over here very clearly. So here there are 4 options that we come across, whether the statement A is right, B is right. If R is the reason for assertion, is the first option there, right? So like that 4 options are given over here. I am going to analyze all these statements now. So meiosis 2 is similar to mitosis. Of course, it is 100% right. So statement A, that is assertion, is right. And reason: meiosis 1 cannot occur in haploid cells. So meiosis 1 cannot occur in haploid cell. Of course, it's also right one. Then so analyze the answer there yourself. And the next question I am going to start with. Embryo formation is lacking in thallophytes. Thallophytes are the lower organisms in which embryo formation doesn't occur over here. So non-embryo phytes they are called as. Hence in thallophytes it doesn't occur. It is the right statement. Assertion is right. Meiosis occurs in the zygote of thallophytes. Because of this reason only meiosis occurs in thallophytes. So especially in zygote. So that's why, so option A as well as R both are right over here. R is the reason for A. That's why, so option A will be the correct answer for this question. Next one, DNA replication does occur between meiosis 1 and meiosis 2, he says. So DNA replication always occurs in the S phase of interphase. It doesn't occur between meiosis 1 and meiosis 2. That's why this statement is wrong. Assertion is wrong. Whatever the reason, in meiosis nucleus divides once but chromosomes divide twice. So this is also a wrong statement over here. So that's why both A as well as R are wrong over here in this case. Next one is the last question. Assertion: mitosis is called homotypic division. Of course, in mitosis already we have discussed 2n number of chromosomes are present in the parent cell and the same number of chromosomes are maintained in daughter cell also during mitosis. Hence equal number of chromosomes are seen both in parents as well as daughter cells. Hence it is called as homotypic division. So mitotic division itself is homotypic division. And meiosis 1 is also meiosis 2 is also homotypic one, but meiosis 1 is heterotypic one. So second statement reason will go through. 2 daughter cells are quantitatively as well as qualitatively identical over here. So in homotypic division, so in both the daughter cells, quantitatively as well as qualitatively both are similar to the parents. Of course, it is 100% good reason for assertion. That's why option A will be the right answer. Both the statements are right and R is the reason for assertion. So thank you. By this we finished all the questions of this chapter now. Thank you.